Okay, so we're very privileged this morning to have Dr. Brian Ballios with us. Dr. Ballios obtained both his Doctor of Medicine, MD, and Doctor of Philosophy, PhD, at the University of Toronto in the combined MD-PhD program. His doctoral work focused on new approaches to the transplantation of stem cells and their progeny for the treatment of retinal degeneration. Dr. Ballios developed the first injectable hydrogel-based delivery system for transplanting stem cells into the retina. After completing his FRCSC in ophthalmology at the University of Toronto, he undertook a subspecialty clinical fellowship in inherited retinal disease at Massachusetts Eye and Ear and Harvard University. So we're very privileged to have him. He is going to be speaking stage right, so if anyone wants to move over to the right-hand side, like we were doing yesterday, please help me in giving him a really warm welcome. Thanks so much for being here.
Thanks, John. I heard that David Gam gave a great talk and stood over here, so I thought I would not try to buck what he had done. He's a very intelligent and smart guy and very astute, so we'll give it a shot. My slides are on both sides, I think, and I should be able to advance them from both sides, but if anyone needs to move, that's fine too. I have a few slides, but I want to keep it very descriptive as well. And I really thank the organizers of the meeting for inviting me to talk. I'm an inherited retinal disease specialist, and I work in Toronto, Canada. You know another inherited retinal disease specialist, Ajoy Vincent, who is here this weekend as well, a colleague and friend. And as John said, I did my fellowship training at Massachusetts Eye and Ear with Dr. Rachel Huckfeld, Dr. Eric Pierce, and Dr. Jason Commander, and it was a wonderful experience. And I've met some of you in this room, and that's been really great. And we have a lot of patients in Canada as well who have choroideremia and are searching for cures and treatments. And I see a lot of them in my clinic as well now. And I have a laboratory where I study stem cell biology. And my interest in stem cells goes back over 10 years to when I was an undergraduate and starting in my PhD sciences. And I became very interested in stem cells in the retina because the retina is a very organized tissue, as you'll see in my presentation. And we can really image the retina quite amazingly in patients with clinical disease. We can see this tissue. And because we can see it, we think it's a wonderful target for new therapies and regenerative medicine. And I think the eye is leading the way in this respect in terms of all body systems, including everywhere in neuroscience. So I have some disclosures that are not totally relevant to the content of this talk, but I've been involved in a number of clinical trials as an investigator and sub-investigator, and I have funding from some sources and health charities that help me accomplish my work. Now, just start by, we're talking about the retina and talking about human biology and the retina as a target, and just to remind us that genetics in inherited retinal disease really works to our advantage. In a lot of medicine and other hereditary body system disorders, they have very complex genetic inheritance patterns that are not necessarily easy to deconvolute and to find the causative genes. But we're very lucky in retina in that the vast majority of retinal inherited diseases, including choroideremia, follow the basic Mendelian genetics. And Gregor Mendel was the pioneer of modern genetics by identifying different inheritance patterns in terms of how genes are passed between generations. And as we know, the choroideremia disorder is passed in an X-linked fashion. And the vast majority of inherited retinal diseases, of which choroideremia is a part of that family, follow the classic Mendelian inheritance patterns. And that's allowed us to identify the genes and allowed us to study these disorders and create animal models. And as we'll see, it's been to our advantage, but also challenging in some disorders, including choroideremia, to develop models that help us deconvolute these disorders. But again, inherited retinal diseases as a group are, while being inherited in a Mendelian fashion, are very heterogeneous in terms of their genetic underpinnings. And there are over 250 different causative genes that have been identified for genetic disorders. And so it's wonderful when we can identify a new gene that causes a person's retinal disorder. And you can see choroideremia on this slide off to the right because it does really represent a unique phenotype. But for other genes in inherited retinal disease, sometimes it can be confusing as to how the clinical presentation of the patient in terms of the symptoms they're experiencing and how their retina appears correlates with what genes are involved. And so this is a growing science as well. And finally, I think it's worthwhile noting that we really live in the age of molecular medicine. And prior to 2001 and the mapping of the human genome, it's really quite difficult to identify genes involved in inherited retinal disease, and hard to identify for any individual patient entering the clinic exactly what gene was causing their disorder. Of course, choroideremia presents with a very distinct presentation in terms of how it appears in the retina. But I think we should remember that prior to about 2007 or 2008, the cost of sequencing a human genome was well over $10 million. This was really relegated to the areas of research and research funding. This wasn't really accessible to the patient coming into the clinic every day. And as we've seen developments in technology, for example, in next generation sequencing and our technology at mapping and sequencing the human genome to discover new disease causative variants, we've been able to bring this cost per sequencing a human genome into the realm of clinical possibility, where we can actually do this feasibly with a patient in the clinic. And commercial sequencing, you see, really comes on board just in the last 7 years or so, around 2015, where the cost per genome comes down to around $1,000, which really makes it able to market this directly to the consumer being the patient and the clinician, in this respect, being able to order a genetic panel to discover a gene. So this is going to, I think, help accelerate how we find different causative variants and discover patients' genetic causes of their disorders. And it's going to help us for patients who haven't been able to find a gene, and also to get patients who present with choroideremia and other inherited retinal diseases to a genetic diagnosis faster, which is going to help us with both research and treatments. This may be information which all of us know. The retina is composed of light-sensitive cells, rods, and cones. We all know about rods. Rods outnumber cones in the retina. There's over 120 million rods in the retina, and they're responsible for our side vision, our night vision. And they really don't function much in the daytime, but they're responsible also for helping us in dim light conditions. In contrast, our cones are about 5 to 7 million cones per human retina. They give us our central vision, our color vision, our high visual acuity, and our vision in bright light conditions, and they help us adapt between rapidly changing ambient light. And if you've been to your ophthalmologist recently, then you may have seen pictures of the inside of your eye, or you may have heard them describe these pictures. And here I show some photographs, and this is how when we look at an eye, a human eye, this is the first thing we see. And so we often take color photographs. Of course, the eyelashes sometimes get in the way. That's okay. And we have an optic nerve that connects the eye to the brain and blood vessels, which snake out across the retina. And when we analyze these photographs as retina specialists and as inherited retinal disease specialists, we're very particular about what we look at, and we're very picky and critical about what we see, because we're always looking for the earliest signs of symptoms and problems when a patient presents to the clinic. And when I look at this photograph, I see optic nerves that have a healthy color and appearance and blood vessels in a normal pattern. And I also see a normal and regular pigmentation to the retina. Sometimes we also supplement our clinical examination with black and white photographs using the autofluorescence, or the natural fluorescence of light that reflects back to us after a bright green or bright blue flash of light in the eye. This creates some characteristic pictures that we also analyze. The nerve and the blood vessels are always dark, but the retina that sits behind them shows up as a light gray color with a dark smudge in the center for a high acuity vision. And these photographs can be very, very helpful for following the progression of conditions like choroideremia over time, because they show us subtle changes that sometimes don't show up in the color photographs, or which may be obscured based on the different coloration of a person's retina. And then what I think is really exciting is that even in the clinic, and this is one thing that drew me to ophthalmology, is we can almost see the individual cells in the retina. And at the top here is is a tissue section of human retina, like I might see in my lab when I'm doing research. And we can see all the little cells lined up, light-sensitive cones in the center and rods out towards the side of the human retina, and all of the helper cells that help them do their business that connect those light-sensitive cells to the brain. In the clinic, we can take the same kind of photograph using a technology called OCT, optical coherence tomography. And that OCT photograph doesn't allow us to resolve the individual cells, but it does show us quite clearly the layers in which they live. And I draw your attention on this photograph to a long dark band that you see in that. And that long dark band is where the light-sensitive cells live. It's where the cell bodies, we call them, of the light-sensitive cells live. And so you can look at the top photograph and the bottom photograph and see that the layers, you can make correspondence between them. And when we see some of those layers thinning or disrupted, then we understand that the anatomy of the retina is disrupted. So here's a photograph of a typical choroideremia patient. This one taken from a young man, age 7. And we see, when I look at these photographs, that the optic nerve has a nice normal color. So the signal getting to the brain is not disrupted by the disease. The retinal vessels have a normal appearance and pattern. And the center of the retina appears dark, as it should. This patient has 20/20 visual acuity, but when we look in the periphery, we see that the pigment is not as even and smooth as we might expect. But it can be difficult, even at this stage, to determine, well, how affected really is the function of this retina? And are there areas that are more thin or less thin? And that's where the autofluorescence photographs become very, very helpful, these black and white photographs. They show us areas where we can see the retina is quite thin in these pockets of dark in the mid-periphery, while the center remains bright, where the retina is thicker and the patient maintains strong central vision. And over time, those areas in the periphery coalesce and become more prominent, and the center area is relatively spared until later in the process of the disease as years and decades of life go on. Just waiting for my next slide. Yes, and here's an OCT image. So there was another copy of that OCT image I showed at the top. So here's a patient in the anatomy we might expect in the center. And what we see in choroideremia is that if you were just to zoom in on the very center and ignore the periphery, you'd see that the anatomy in the center has all the same layers that it does in the normal anatomy. And so patients have maintained very strong and normal central visual acuity. But if you move off this hill or this island of vision, and we see that the layers of the retina, particularly the outer retinal layers where the light-sensitive cells live, become thinner. But it's not just the light-sensitive cells that become thinner, it's also the blood vessels that nourish them underneath. The blood vessels from a layer called the choroid, which is where choroideremia gets its name from. And the earliest physicians looking at this disorder recognized that those blood vessels were thinning. And they thought that perhaps this represented the key element of the progression of the disease, that it was some progressive loss of these blood vessels causing thinning of the outer retina. We know from molecular genetics and the gene, the choroideremia gene, REP1, that the story is a little more complex than that. And so the focus of my talk today will be on how can we try to rebuild the retina using stem cells, because these are areas where the light-sensitive cells have gone away over time. And while gene therapy is targeting a way to enhance light-sensitive cells and their supportive cells, we need a way for these patients to rebuild the parts of the retina that are lost over time in disease. Well, just for some context, the patient's mother, who's asymptomatic, also has very prominent pigmentary changes in the retina, but has normal visual function and normal visual fields and has no concerns with their vision, which is also one of the very interesting features of choroideremia, in that in the carrier state, retina has such a prominent appearance compared to the normal appearance of the retina, and yet has a completely normal function. Well, I talked a little bit about gene therapy, and gene therapy is an evolving science, and we know about, in this room, I'm sure many of us know about the the exciting developments in gene therapy and many groups that are working on it for choroideremia, also some of the setbacks and challenges that have been had in the field. Those approaches have focused on a technique called gene augmentation, which is about trying to put back into the retina a normal copy of the choroideremia gene. And it's a great science, and it's been helpful for some retinal diseases, and it's had challenges in others. But it also leaves behind some patients with inherited retinal disease who don't need gene augmentation, but rather need editing or changes to the gene to cause proper functioning, not because they have too little of the gene, but maybe because the gene doesn't have the proper spelling. And we know of several gene therapy trials, some of which are underway, some of which have not met their primary endpoints, some of which are recruiting new patients right now. But I think the challenge is in gene therapy it's all about timing. It's about trying to get to patients before they've lost the majority of their photoreceptors to preserve and enhance the vision from the photoreceptors they have. But it doesn't really represent a regenerative, I would argue, approach to vision loss, where you're looking to put back light-sensitive cells. So what are you going to do for this patient who has at the center of their macula, the very center of their retina, a small area of bright signal on the autofluorescence that represents maybe 20/30 or 20/40 visual acuity, but is severely limited in terms of the utility of that vision, in terms of how they can navigate and find their way around? How are you going to put back the light-sensitive cells in the large areas that surround that area, not just to protect it, but to bring vision back? This patient has small visual fields, as you can see, less than 20 degrees. So my group and a few other groups, and maybe David Gam talked about this a little bit yesterday, we believe that stem cell therapy may represent a restorative or regenerative approach. The hurdle's a bit higher for stem cell therapy because it's a living therapy. It's about putting in living cells into the human body. This is different than designing a new drug or molecule to give as a pill that can help enhance the function of a body system. This is about introducing new tissue. So what is a stem cell? A stem cell, as a definition, has a very rigorous definition. When we think about stem cells, they can come from many places, but they all have to show two cardinal features in common. The first one is that they must have the ability to self-renew. That means when a cell divides, when a single cell splits in two, a stem cell, if it's a stem cell, must make another copy of itself. That means a stem cell is an immortal cell. It always regenerates another copy of itself, and the second cell splits off to become another progeny. So a stem cell must have that self-renewal capability. The second cardinal feature is that it must be multipotent. And to unpack that term, multipotent means that the progeny of that stem cell, as it divides, must be able to make multiple different cell types in the human body. So if we're calling a cell a stem cell, it has to have those two properties. We've probably heard of multiple types of stem cells. There's embryonic stem cells, which we can isolate from the developing embryo, and which many labs had done that decades ago. And because they're immortal and continue to divide, they continue to grow in laboratories around the world. And they represent a plentiful source of cells because these stem cells are never exhausted. They always divide and produce new cells. We can get stem cells from adult tissue, retinal stem cells, for example, that are resident within all of our retinas, that are quiet as adults. We call them quiescent, meaning they stop dividing when we grow up and our eye forms. We've discovered ways to isolate these cells from adult tissue, from donor tissue, after patients pass away and they donate their eyes for research. We've been able to find resident tissue-specific stem cells within the retina that we can wake up and get to divide again. The eye has a remarkable ability to form, and then as humans, we want to suppress the ability of cells to proliferate or divide uncontrollably because those processes typically create cancers. And so our body has a lot of checks and balances to keep cells from dividing as we grow up. These retinal stem cells that we isolate and grow in the plastic petri dish can represent a source of different retinal cells, including photoreceptors, that I've spent a lot of time researching. And then you may have heard of induced pluripotent stem cells, IPS cells. And this technology came about because of the interest in trying to find new sources of cells that didn't make use of having to isolate human embryos to find stem cell sources. And so an induced pluripotent stem cell is a very special kind of stem cell that we can make from an individual person, an individual human, or even an individual patient. And iPS cells have a lot of promise for not only treating disease, because they're isolated perhaps from the same patient who has disease, or perhaps from another patient who's closely related who doesn't have disease to generate tissue, but also as scientific models to better understand disease, which is some of the topic of the work in my laboratory. Because for some inherited retinal diseases, as I mentioned earlier, animal models or other models in other species that perfectly mimic the human situation don't exist. Well, let's take a dovetail for a second into what stem cells have been used so far in terms of therapy for inherited retinal disease. And while there's no specific stem cell trial right now for choroideremia, there have been various stem cell studies, and some that are ongoing, for inherited retinal disease, which involve the loss of photoreceptors, that we think progress in these trials will help us understand how these therapies might be useful if they're ever approved for choroideremia. And so in this particular slide, I borrow data from a colleague of mine, Dr. Michael Young, at Harvard's Skepin's Eye Research Institute, who has published this data with his colleagues in 2014 and 2015, showing that these retina-specific stem cells, when transplanted into an animal retina, a rat retina, are able to survive and associate with that retina, and some of them seem to have the ability to actually extend little processes and connect to the existing host retina. This was very exciting data that they had published. If you look on the right in the micrograph, the transplanted cells, they put a green fluorescent label on them so they could follow them once they'd been transplanted. And they could see these green cells next to the blue host cells, and even some of the little green cells migrating in to sit amongst the blue host cells, and that was really exciting. They did that in a normal animal retina, and they also did it in an animal retina from a rat that suffers a genetic cause of inherited retinal disease. And they showed that these cells could associate. And so that led to a clinical trial. A clinical trial for injecting underneath the retina these retinal stem cells, with the hope that in these inherited retinal conditions, and in their trial they used retinitis pigmentosa, the hope that these retinal stem cells would do what they did in the rat. They would associate with the retina, and they would extend little processes and maybe replace the function of those light-sensitive cells. And I'm showing a video of these cells being injected underneath one of the blood vessels in the retina. You can't see the cells. It's a clear solution. And it's hard to even know that the injection is actually going on unless you have a microscope to see. But afterwards, after the injection is done and the surgery is complete, you can see these areas that we've outlined in red on the slides of where the cells are sitting. And they look just like the cells would look in the plastic petri dish. We had injected them onto the surface of the dish. They sit there underneath the retina. And the hope in this trial was, well, maybe they'll do something, right? Maybe they won't just sit there. Maybe they'll connect to the existing retina. We started out with patients with very, very low vision, patients who had difficulty reading the eye chart, or even had difficulty counting fingers on the hand in the clinic, and showed excellent safety of this injected stem cell material. At the phase 2 clinical trial, where they say, well, okay, safety is good, but is it giving some efficacy? That has yet to show the efficacy signal that the investigators are looking for. They've tried moving to patients with stronger baseline vision and more enhanced retinal anatomy, but have yet to see any efficacy signal. That was a bit of a disappointment for the investigators in this study, of which I was one, because we were really hoping that we would see some of the function that might have been suggested in the animal models, even though it wasn't exactly the same animal model of genetic diseases we were using in the humans. But this was the first time this had been done in humans, and so it was exciting to learn about how that these cells can actually be well tolerated in people and actually be injected underneath the retina without causing too much disruption to the retinal anatomy. And so the safety signal was excellent to see because we never know with a new cellular therapy if it's going to be helpful. But what I'm going to focus the rest of my talk on is also the development, I think, of cell therapy and stem cell therapy 2.0, which is saying maybe we're not thinking about using stem cells themselves, which we're not telling what kind of cell to become, a rod cone or a rod photoreceptor or a cone photoreceptor. But maybe we need to use the progeny of those cells, the rods and cones, to replace the light-sensitive cells. Maybe that was the challenge here, and there is some data in the lab to suggest that that's the case. Before I move to that, I'll just mention that there are several clinical trials going on to replace some of the helper cells that are disrupted in other retinal conditions and inherited retinal conditions, like Stargardt macular dystrophy, for example. And Stargardt and choroideremia share one common feature in that both patients with choroideremia and with Stargardt lose the retinal pigment epithelium, the pigmented cells that support the photoreceptors inside the eye. And so replacing those pigmented epithelial cells will be important for any therapy, regenerative therapy in choroideremia. And so there have been regenerative patches developed of pigmented RPE cells that can be grown in a plastic petri dish from those stem cell sources I mentioned before. And those are being transplanted in patients with both Stargardt disease and also dry age-related macular degeneration. The hope being that we can show that those pigmented RPE cells can be well supported by the host retina and be able to regenerate those areas of RPE that are lost. You know, in ophthalmology, we have a wonderful tradition of finding new therapies and then co-opting those therapies into other similar conditions. In age-related macular degeneration, this is how we developed these anti-VEGF therapies like Lucentis or Eylea, these injectables that we use for wet macular degeneration. The very first one of those was a drug that was actually developed for colon cancer to suppress the growth of blood vessels. And some brave ophthalmologists and some brave patients decided to try using that drug for a completely different purpose, to try to suppress blood vessel growth inside the eye. It shows you that once a drug is approved by the FDA, once it's approved for use in one particular disease, it can often be explored for its use for other indications. And so our hope in cell therapy is that while one very specific disease, like for example Stargardt disease for RPE replacement, while that might be the basis of a clinical trial, if that therapy is proven effective there, it might be tried in patients with other types of retinal disease, including choroideremia. So I think it's important not to just focus on the research in choroideremia, but think a little more broadly when it comes to stem cell therapy about what's being done in the retina in general and what things are entering clinical trials. So we're very excited to see the results of this work, and the early results have shown good tolerance of the eye for these pigmented cells, that they can survive for months and months after implantation, even upwards of over a year in some of the patients who were transplanted at the earliest stages. And they can even be made from the patient's own cells or from living relatives who don't have disease. For the last part of the talk, I'll focus on some of the research I'm doing in the lab on cell therapy 2.0. And I'll just start with this sort of fundamental question that my lab is asking, which is maybe how can we buck the traditionally approached questions, the traditional ways that we approach questions in retinal disease, and including what you might classify as rare retinal disease, though it's not rare to anyone in this room, like choroideremia. Well, the traditional models we've used in the lab to explore these concepts are cell culture, which takes place in the plastic petri dish in an incubator. Animal models, I show a little mouse here, and because mice and rodent genetics are very malleable, you can change their genetics to try to express or suppress certain genes to try to study disease. And ex vivo, getting human tissue to try to study the cells also in the plastic petri. Oh, I'm back. Also in the plastic petri dish, also in the lab, ex vivo. Well, in my lab, we tried to take a lesson, a page, use our strategy, a page book from the developing retina. We know during retinal development that the cells that are born in the retina, the rods, the cones, and all of their helper cells are born in a very, very regular and prescribed and regular temporal fashion. And so some of those cells are born early, like ganglion cells and horizontal cells. These are helper cells in the retina. And some of them are born later, like the rods. Interestingly, the cones are born much earlier than the rods. And so this process happens in a very regular and reproducible manner, okay, across human development and across mammalian development. And so we've been asking in our development of stem cell 2.0 therapy is how can we use the different factors that are at play in development to help us make stem cells become the kind of cells we really want: the photoreceptors, rods and cones, and the RPE cells. We've been able to ask questions such as maybe the cone photoreceptor might be the default state for a cell that is born from a stem cell. There's some evidence in development. We've been able to show that perhaps this is also true in our plastic petri dish when we're growing cones from stem cells. We've been able to identify factors that can make very, very pure cultures of rod photoreceptors or cone photoreceptors. We think that these are the cells, not the stem cells that were used in some of the clinical trials I presented earlier. We think it's these cells that perhaps are going to be the most useful for therapy because these are the ones that the retina really needs. And these are the ones perhaps we should be transplanting if we want to regenerate those layers. As I showed you in that OCT photo of choroideremia, it's not the helper cells that are lost. It's not the ganglion cells that communicate with the brain through the optic nerve. It's not the bipolar cells that are lost in choroideremia. It's the light-sensitive rod and cone photoreceptors that really give the challenge and the final common pathway. So we need to find therapies where we're replacing those cells specifically, not just trying to put back a stem cell and hoping it knows what to do inside the eye when we put it there. And we've used our research to find these various cocktails of factors that allow us to transform a stem cell over time into a cone or a rod. And it's even allowed us to identify new factors, for example, transcription factors that govern how these cells change their state from stem cells to rods and cones that help us to identify the right stage at which to isolate these donor cells for transplantation. And so we're finding things that scientists have never known before about the way cells transform from stem cells into these specific rods and cones. The goal always being to find what is the proper donor cell population that's going to interact with the adult retina to replace light-sensitive cells. This is some science and some staining on some slides where in the plastic petri dish, as we grow these cells, we're able to find the specific markers of rods and cones and even some novel markers that help us identify the correct stage at which to isolate these cells in the dish for our cell transplantation experiments. And in my lab and in my research in the past, we have done cell transplantation, and we continue to do it even now. We label these cells in green, just like Dr. Michael Young did in his experiments, except in this time we're not labeling the stem cells, we're labeling the photoreceptor progeny themselves and doing transplantation. We can see that some of these cells do associate with the host retina and with those host photoreceptors, and even some of them migrate in and take up residence amongst them. And that's really exciting results. We collaborate with bioengineers. My background as an engineer and a chemical engineer, I worked for several years designing new biomaterials to try to help transplant these cells and help them engraft in the retina. Because we know that if we just inject some cells willy-nilly into the retina in a salt solution, they tend to stick together and then expire without actually integrating into the retina. But if we use a biomaterial matrix, we've shown in our work that we can actually get them to survive for longer and have a more healthy appearance and express all the genes we'd like them to express. And we think that made some sense because the retina is composed of not just cells, but also a tissue matrix. And so if we designed a matrix, we were able to show that these cells would survive for longer. And not just that, but work out of one of my colleagues in my research institute, as well as others overseas, like Rachel Pearson in the UK, have shown that these cells that are transplanted don't just take up residence inside the retina, but sometimes they actually pass on genetic material between the cell and the host retina. And so we think they might also be a vector for gene therapy. You know, a diseased retina lacks a specific gene or a gene product, like in choroideremia, the REP1 gene. And so a transplanted healthy cell mass may be able to transfer healthy copies of that gene to host cells, and we think this could be really very helpful. And then we're exploring this together with our colleagues as a potential mechanism for how cell therapy may work, not to just replace cells, but also support the healthy functioning of those cells in the retina that remain. With my final 3 slides, I'll say that we're not happy really with any of these models in general, and these traditional laboratory models, and how they limit our ability to approach questions in rare retinal disease. And I think our field in vision science is experiencing a revolution in terms of how we think about how to actually explore retinal disease and retinal conditions in the lab. Static cell culture, which is in a plastic petri dish, has suffered from the fact that there is no tissue around those cells in the petri dish that mimics what these cells are actually living in the human eye. We can only study usually single cell types at a time, and there's no immune system around to modulate the life and growth and cycle of these cells. So these cultures are also ultimately limited in how well they replicate the human condition. Well, animal models seem obvious in that they don't really replicate the human condition, even if they are other little mammals like the mouse. The retinal physiology and the structure of a mouse retina is completely different than a human retina. It has different ratios of rods and cone photoreceptors. They don't respond with the same rapidity that our rods and cones respond to light. They have different, what we call electrophysiologic properties. And mice also don't really have a macula, which is a really important feature of a human retina for our high acuity central vision. And of course, there's some who have ethical concerns over the use of animals. So there's a number of challenges using animal models. And then ex vivo, getting tissue from patients and donors for study in the lab is really limited by the short lifetime. It's hard to keep this tissue alive in the lab, and limited availability, of course, especially when studying rare conditions like choroideremia, for example, where you might not get eyes from a donor patient with the choroideremia when they pass away very often because the disease is not very prevalent. And so that's where we've turned to iPS cells that I mentioned before that we can make from any patient to try to study disease. And so in development, the eye develops from the brain in a very regular pattern, in a very regular and predictable way. And some very intelligent scientists that we've worked with in the past have shown that we can use the same sort of chemical signals in the dish to get unorganized groups, disorganized groups of cells and stem cells to actually self-organize and start to grow little retinas in the dish. And these are mini retinas that are about 0.5 millimeter to 1 millimeter across in size. They're very tiny, but they're growing rods and cones in an organized way, just like a normal human retina. And that for us is really exciting because it actually mimics the structure that we see on an OCT scan of the human retina. And we see rods and cones that we could study. And we can isolate those cultures from patients who have specific genetic misspellings in their genome that cause retinal disease, like choroideremia. We call these little structures organoids. They're complex, stratified tissue, and they have most of the major retinal cell types. There's no ethical concerns with their use. They're isolated from adult patients and grown in the petri dish. And the concern is the research is expensive, I guess, perhaps. They are long-living and can be kept alive in the dish for well over a year in cases of careful culturing. And they mimic the complex interactions between cells that we see inside the human eye. And in my lab, we've been able to derive these from patients both with retinal disease and without retinal disease. And we're working on trying to derive some from choroideremia. And what we want to do is explore and understand what's different about the rod and cone photoreceptors fundamentally in patients with choroideremia and other inherited retinal diseases. I think one of the challenges for cell therapy in choroideremia and we'll probably explore this, I imagine, in the question period, someone will say, well, what about those blood vessels that go away? Won't you need blood vessels to help support the survival of cells afterwards? And I think that's a valid point. I think at the end of the day that any cell therapy for choroideremia is going to depend not just on replacing the rod and cone photoreceptors, but also on replacing those pigmented epithelial cells and also finding a way to enhance blood flow and physiologic nourishment of those cells in the plastic petri dish. But I think we also need to start to understand, how do those donor cells interact with the host tissue, specifically in patients with choroideremia. And the hypothesis in my lab is there's differences between patients with different retinal diseases that mean that not all cell therapies will be of equal utility for every single patient. And the better we understand how that interaction plays out, the better we'll be prepared to introduce these cell therapies to the clinic. So my last slide, conclusions. Inherited retinal diseases like choroideremia are becoming actionable diagnoses because there are many new actions we can take, even if they're research actions to understand the disease better. So access to timely genetic testing is critical, and I mentioned how the cost of genetic testing has come down. So it's important that we try to get a genetic result for every single one of our patients. Gene therapy is probably most useful while there's light-sensitive cells around to rescue the function of. And cell therapies are what we think of as being very promising when photoreceptors and light-sensitive cells are gone. And this I put in small font, but really it's the big question in my lab. Should we really be ignoring the specific genes that are important in cells? Or are there gene and disease-specific effects? And I would argue that my hypothesis is there are very specific effects that cell therapies can have in specific types of genetic disease, like choroideremia or other specific genes affecting IRDs. And so I think understanding and finding therapies will involve these new models of disease like we're working on, iPS cells in our lab, and of course new models of care and clinical research in terms of how we approach patients as ophthalmologists. And when I speak to my colleagues in ophthalmology, I really try to emphasize some of these top points, especially this actionable diagnoses, that patients who have choroideremia be referred on to specialists in inherited retinal disease to really understand their condition and their disease fully, so they can be prepared for these next generation of gene and cell therapy. And so I'll finish there because I want to leave lots of time for questions. I'll leave my acknowledgments of the many talented people I've worked with in the clinical service and in my lab that's growing over the last year, and we have another graduate student here starting that I didn't put here. My sources of funding. And that's good. I'll stop there.
Thank you so much, Dr. Ballios. Let's give him one more round of applause. All right, fantastic. So I think we're now open for questions. And I think we'll have some people with mics in the back. So if you have any questions, please feel free to raise your hand and we'll get you on mic.
Test.
Please try and talk into the mic so we can get it on camera for those who are watching the streaming content. Thanks so much. Any initial questions?
That's a joke. Thank you. That's becoming the joke. I always have a question. So the first thing I wanted to ask is not really a question, is I'm not sure I understood, and I would like to reframe what I understood. So with the stem cell 2.0, are you saying that, first of all, we're going more and more specific and moving from injecting, let's say, the mechanical piece, instead of doing that, it would be focusing on injecting the growth, the fuel for the growth development? I don't know if I understood.
I think you're quite close. So, and it's a great question, and it really hinges on what we always thought stem cells could, and what we're discovering they can and can't do. It's about becoming more specific about the therapy, as you say. When we, and I use the big we, a collective group of scientists in stem cell biology, when we began to understand stem cells and how we could, and the types of cells they could become in the human body, including retinal cells, when we started to understand that better and better through the late 90s and early 2000s, we had this incredible optimism that stem cells were this panacea, that they could cure all conditions because they were intelligent cells that could become any cell type that was lost. And it turns out that they are pretty smart, but they're perhaps not as smart as we give them total credit for. They don't necessarily in our transplant models turn into all of the types of cells that we want them to. Sometimes they stay as stem cells. Sometimes they don't transform into photoreceptors. Sometimes they transform into the other cell types that are not lost in disease, not the ones that we really want. And so we've been looking as a field, again, looking back at stem cells and saying, well, maybe we were asking a bit too much when we thought that maybe the stem cell would know what to do just in isolation within a transplant. Maybe we should be a little smarter about it and use stem cells as a source of the useful cells we know we need in specific diseases. So the rod and cone photoreceptors, for example, or the RPE cells in those patches. And so a lot of the focus of Cell Therapy 2.0 is about taking the stem cells as a source, finding the right source of stem cells, and finding ways to create the best population of donor cells. Maybe they're not fully mature photoreceptors, maybe they're immature photoreceptors or immature pigmented cells, and use those as the cell source for cell therapy and give the retina what it really needs. And we also wonder if maybe that's been part of some of the challenges that some of the stem cell clinical trials have encountered to date, in that we haven't seen in those trials the rebuilding of those layers of the retina that we originally thought we might expect stem cells to be able to do. So that's the focus, and you're right, it is becoming more specific of what cells we need. And as we become more specific, the research I'm exploring in my lab and what we've noticed is that in specific animal models of specific genes involved in retinal disease, those progenitors don't all behave the same. They don't all engraft the same or survive the same or associate with the retina in the same way. And that was really interesting to us because we thought, well, when you lose photoreceptors, it must be the same in every condition, right? You've lost the cells, so you just need to replace them. But I think it's a lot more complex about how these cells interact with specific retinas from depending on which genes have misspellings in them. So that's why we think those, in the final slides, those models are going to be very helpful.
I'm going to add a quick follow-on question while we get the next one queued up. So I was curious, and I thought that was fascinating. I was also getting lost in thought a bit, so I apologize if I missed this. But so if we take the progenitor cells and we further differentiate them, and then we try to install them appropriately, can you speak to a little bit more about the idea of recreating that milieu that will help with the installation? So could we kind of augment the chemotactic factors to try and get them to make the right connections to the old cells?
Yeah, great question, John. I painted in broad strokes, but that's our hope, is that we create something that's, through our biomaterial-based approach, something that's much more akin to an extracellular matrix that has the right factors within it that support cell survival and function. And so with that gel that we showed in those early pilot studies was very helpful for cell survival. We were able to uncover what parts of that gel were helpful in their interactions with the cells. And we've even been able to attach other growth factors into that gel to help maintain cell health and differentiation after transplantation. So we've been focused on transplanting immature photoreceptors, but we don't want to just depend on the retina to tell them how to mature. We want to have the factors around within that matrix that will tell the cells after they've been transplanted exactly how to mature and become the photoreceptors that we all use in our retina. So it's I was painting with some broad strokes there, but that is the exciting aspect. Now the question is, do we need different concentrations of those factors in different retinal diseases? And that's where the IPS model comes in, because there's not great animal models of choroideremia, for example. But you can isolate these IPS cells from patients with choroideremia, grow these retinas in the dish, and we want to use them as a model of cell engraftment in the petri dish for specific diseases and specific conditions, and get around the idea that the limitation that we all talk about. We don't have good animal models of disease, and so that's an exciting way.
Fantastic, thank you. All right, who's next?
Okay. Why not? Hello. I'm a Canadian. We're a couple Canadians here. Over the years, we've been seen by, thank you, Dr. Ion at Sick Children's Hospital. Some of our kids have as well. So Are Dr. Young's files accessible to you? Should we or they as adults be seeing you over time, both carriers and affected males?
Oh, thank you for your question, and thank you for coming. And it's nice to see more Canadians here too, as a fellow Canadian. We should have booked a bus. But the short answer is yes. At least in our healthcare system, once a patient is booked to be seen in consultation, our healthcare institutions are, as most healthcare institutions are, quite protective about where their records go, especially from the children's hospitals. But once a patient's been once a new provider is within the circle of care, then those records should be accessible. And Dr. Hayon and I, as well as Dr. Vincent, who spoke yesterday, work very closely together in the city and have been trying very hard to develop a citywide model of care in Toronto, which really translates into an Ontario province-wide model of care, to share patients with these rare conditions. To break down those silos that have traditionally existed between institutions and made it difficult for patients' records to get back and forth. And I think as providers, we understand the importance of not just seeing the patient and getting our own photography and imaging and what we see on the day, but also getting access to those records and investigations that have been done in the past. So the short answer to your question is yes, I think that's possible, and we've been able to do it for others.
Other questions?
What is your
Yeah, I'll repeat the question. I think the question was, what's number one on my wish list? Do you mean it with my lab research, or in general for aspirations for a cure for choroideremia? That's my number one wish. Specific to the lab, well, so one of the things we're really interested in these IPS models is exploring how these cells that we're going to be transplanting engraft in real time. It's not enough to put them together and take a photo at 1 day and a photo at 2 weeks and a photo at 3 months and not be able to watch how they interact over time. And there's new microscopy techniques that have been developed, live cell imaging and light sheet microscopies, and I won't talk about the specific models, but having access to one of those light sheet microscopes, which is a very expensive piece of technology, is at the top of my wish list, because I really want to understand how these cells interact early on and persistently over time. And so if you were to ask me, that would be one piece of equipment that would be very helpful in the lab.
Doctor, thank you for your information about the building of the iPSC models. My question really is just a short one. How long can those organelles survive? I know they're very young cells, but how long can they survive, or are there factors that you put in to enhance or speed up the phenotypical changes within the RP and photoreceptors?
So we're able to keep the IPS cultures alive for well over a year with very careful feeding and monitoring and constant gardening. But they mature over the course of 2.5 to 3 months. And our group, as well as David Gam's group and others, have shown that these organoids may be on an accelerated developmental timeline compared to human development, because of course our eyes don't develop so quickly into maturation over several months. It takes us over 9 months in the developing fetus, and then our eyes continue to develop and mature phenotypically, even postnatally, for years. But in terms of the production of the useful cells and the mature photoreceptors, that at least takes 2.5 to 3 months to see. But they can be kept alive in culture for much longer. I think it depends on the specific disease whether these cells are going to mimic the mature disease phenotype as time goes on. And a lot of these questions are open and remain to be seen. There's been some argument about whether we should be creating these these stem cell banks using gene-specific technologies like CRISPR technology to just create a large bank of disease-specific cells. But I think there's still a lot of value in getting those samples originally directly from the patient, because there's a lot of helper genes that may have specific misspellings in them, along with the choroideremia gene, that make those cells unique to that patient and that specific family, for example. So there's a lot to explore in how these cells mature in the dish.
Question here. If I understood you correctly, you're looking at developing some rod and cone specific cells separately. Could they then be injected in one injection together and then hopefully know where to run when they get there? And then what sort of area do you expect they would spread to? I mean, it would be great if it spread through the entire retina, but I'm going to guess that's not the case.
I think it's a great idea. With the idea of specificity, we want to be able to create very pure cultures, but the idea of mixing them could be to our advantage. And we have the ability to mix them in defined proportions, so 50-50, 75-25, one way or the other. And it really remains to be seen in our transplant models and also in our engraftment models, whether these donor cells have some advantage to being mixed or whether they don't. Now, of course, in the human retina, they're all admixed together. The cones and rods exist as a mosaic. And thinking of a biomimetic retina, we need to be able to replace both in proportions. Now, whether they need to be replaced in the same proportion of 120 million rods to 6 to 7 million cones in that ratio, or is it better to develop a therapy that's more like a 50-50 split? That remains to be, that's an open question in the field. The second part of your question was what area we think they might spread to. I mean, the hope is that we'd be able to treat large areas of the peripheral retina. The retina, as it thins and the outer retina undergoes a process of degeneration, also undergoes a process of tissue remodeling, which may make it less amenable to cellular integration. So we would start with the areas that have been affected most recently and work outwards, so from the macula out and away. But it's a good question, and it's not clear yet what area of the retina should be specifically targeted first. That being said, I think most patients would benefit from enhancements in their pericentral vision, and hopefully, if these cells do what some of our collaborators were suggesting they might do, which is also provide gene products to the cells which are still resident within the retina, then maybe that would be helpful to target those areas that still also have some remaining cells.
I'm going to hop in with one other quick question. I'm just curious. So let's say 5 years from now, you have these gels ready. Do you think that they're stable enough to be injected, or do you think that they need to be placed? Are they rugged enough to withstand, an injection? If you could comment on that, that'd be great.
Yeah, so the gel we developed in that study was an injectable hydrogel. It's a gel which is, I'll borrow the engineering term, shear thinning, which means that as you inject and put pressure on the syringe, the viscosity of the gel under pressure actually goes down. In engineering, we call it the Young's modulus, the ratio of shear stress to pressure, and it actually decreases as it's injected. And so it flows like a liquid through the needle. And once it's delivered and no longer under pressure, it actually forms a hydrogel, more like a jelly substance, you think of it like a thin jello. And we thought this would be very useful because the intrinsic modulus of that gel actually mimics the tissue modulus of the retina. We designed it that way. Other groups have looked at using biomaterial delivery vehicles that are solid wafers, for example, that they've seeded cells on or seeded cells within those wafers to put into the retina. Those obviously have advantages in having a formed structure in which you could potentially laminate or place different cells within different regions of that implant. Think of it maybe like an implant. It's not clear what's the best approach, but we like the idea of trying to, if we're going to use photoreceptors, which need to be distributed as evenly as possible across the surface of the retina, but also need to have the flexibility to associate. We designed a hydrogel that would degenerate within 5 to 7 days after injection, and which would dissolve, but would support them during that critical early period.
So I have another question. You at the very end, you just talked about blood vessels, and you opened a new door for me because I didn't think about that. So how could you tell us more about that? And I guess what I would like to understand, if I understand well, is like if stem cell is all about injecting more cells in order to produce something in an activity, what can we do for the environment and for the channel to nourish and make sure that the cells continue to be well fed?
So that's great. That's, I think, the linchpin question. What to do in choroideremia about the blood vessels? Because cells need they need oxygen, and they need a way to take away metabolic products and everything that healthy blood supply gives us. And that's the big challenge in choroideremia, is that choroid thins. Even if we were to transplant cells, how would they survive effectively and efficiently without an optimum blood supply? And this is where I'll say that my lab doesn't work in isolation. We are addressing some questions with the rod and cone photoreceptors and integration, but there are other groups that we work together with in the community of researchers that are working on using these gel scaffolds, for example, to grow new blood vessels and to replace the natural, what we call Bruch's membrane, the basement membrane that underlies that entire retinal structure, to actually replace it with stem cell derived capillary cells that actually will allow blood to flow through. There's some really exciting data in the dish where they can show that you can coax blood vessel stem cells to turn into capillaries that can seed and can form little channels through these solid scaffolds. And the idea there is to replace the choroidal blood flow, or augment the choroidal blood flow to help with that. That's where I think the challenge in choroideremia, the hurdle, gets a little higher than in some of those, or maybe much higher than in some of those diseases where the blood vessels in the choroid are not lost. But that's not been ignored. It's a big challenge, and it's something that several groups are working on. And I think that at the end of the day, it may be useful to combine all of these cell therapies into one particular transplant. And we are trying that out in the petri dish, to combine these stem cell derived capillary cells with our photoreceptors in multi-layered transplants. So remains to be seen.
All right, help me in giving him one more strong round of applause. We have a final question, I guess.
Sorry.
That's okay.
I had my hand up, but it's
A buzzer beater.
In the extreme periphery.
it does seem to me that in your description, you still have an inception problem. If you use the IPC stem cells derived from the patient, they're still going to have whatever miscoding is causing the problem. You might get a temporary rejuvenation or youthening of the retina, but the eventual underlying problem would just rear its head again in whatever that time frame is. Or if you end up using sort of a known good genetic donor, then you may have mismatch and non-compatibility, and so how are you thinking about that problem? How I realize that's, hey, how do we merge these two ways of thinking of this problem? And it's a big domain, but does that keep you up at night?
Yeah, sort of. That's you articulated perfectly the challenge you would have if you were to derive a donor cell population specifically from the patient's own cells for transplantation, in that you would likely expect, or hypothesis would be, that those cells would perhaps have some function for a time and then undergo the same pattern of degeneration, or some pattern of degeneration. And so deriving the cells from the specific patient with disease is very useful for disease modeling, but not for deriving a cell therapy. But as you said very well, that if you were to use a control population of patients that did not have disease and you derive cells, then you would be always having to struggle with the ideas of donor-host mismatch. That is true in any sort of solid organ transplantation, like kidney transplantation or lung transplantation. Is the idea of that we're all genetically diverse and have diverse immune systems, and that rejection is possible. We have to be very careful of how we match donor and host. So one part of what I didn't touch on is the use of ancillary technologies like CRISPR to do gene correction of the misspelling gene, the misspelled gene that we derive from the specific patient. Just the specific misspelling in the REP1 gene, leaving all the other genes and expression of genes alone. We call this an isogenic line. That means the cell that we've derived is similar to that patient in every single way, with the exception of the one gene that we've corrected with CRISPR. And our hope there is that, well, then because these genes are just the same in 99.999% of their gene and expression, they will be excellently matched in terms of donor and host, because they really will be your own cells with the exception that the gene has been corrected. And we've gotten very good at creating those stable lines. So it only keeps me up at night in terms of how excited I am that we can use those lines that we've created from patients to explore how these cells behave differently in the dish from their mutated counterparts, and also at how we could potentially use them for cell therapy in the same patients. It is about personalized medicine, and these therapies, there's going to be a curve, like there was for gene testing, in terms of the expense of this technology coming in down over time, and the half-life of that curve as it comes down is yet to be established. But yeah, I think there's a way forward to find good donor-host matching by using IPS technology and combining it with CRISPR, for example. We've done it in the lab.
All right, help me in giving him one final round of applause.